Hello and good evening, and thank you to everyone who's attending this evening for our STOPFOP webinar. Just to say hello to everyone, we know we've got people from the UK and further afield. And just if you want to pop in the chat function and say hello and where you're from, that might be nice so people can see who's here. I'd like to welcome our three speakers this evening, Professor Marelise Eekhoff from Amsterdam, Professor Alex Bullock from Oxford University, and Professor Richard Keane from the Royal National Orthopaedic Hospital in London. If you've got any questions, if you can put them in the Q&A section, we'll try and answer them towards the end of the presentation rather than putting them in the chat, because they can sometimes get lost. This webinar is being recorded. And I'll hand you over now then to Professor Bullock. So thank you.
Thanks, Helen. It's a great pleasure to see everyone and to welcome everyone to this. And we're going to tell you today, over the next 40 minutes or so, about an introduction to FOP and the different clinical trials that are running, potentially the impact of the STOPFOP clinical trial and progress there, as well as other trials that are running, and particularly the state of the trials in the UK. So I'm going to introduce Marelise and Richard later, but I'll kick off with a little introduction. If I move the slides, let's go. Okay, so I want to give just a little bit background in the context of, we all know that there's a gene which has been linked to FOP as a causative gene for FOP. And I want to give a little introduction as to, what is it? What does it do? Why is it causing a disease? And then how can Saracatinib in clinical trials, how can this investigative drug have benefit in terms of treating FOP? Marelise will talk about the clinical trial itself, and Richard will expand on that with a wider view of clinical trials and also the sort of what's going on in London. And then we can finish with some questions. So I want to start with some basic understanding so that you know the rationale for clinical trials and what is causing FOP. So I think it's important that you all know the role of the FOP gene. And I hope these pictures are familiar with everybody. So if you take the picture on the left over here, maybe I can put on a pointer. Okay, so we all start off as a single cell that amplifies into multiple cells. And then, we will all then eventually end up, hopefully, with 2 arms, 2 legs, and recognizable head, etc. It's a human body. And of course, the question is, how do these cells in a cluster here become into human form, or if you're a fish form, etc. And it's important also to note that these cells are also constituent parts of us. So your body is entirely made up of cells that would look similar to this. So the question is, how would one cell know to become, I don't know, a blood cell, and another cell become a skin cell, and another one contribute to the toes or the head, etc. And the key point is that all of these cells have to talk to each other. And so the FOP gene, ACVR1, participates in this talk process. So it's part of this communication and signaling such that cells can talk to each other to know what context they belong to. And the way it does this is it acts as a receiver, or what we call a receptor, so that when these messages are sent out, it acts to receive them. And because the human cell has a fatty lipid layer around the outside, the message can't just go straight through the cell. It gets stuck on the outside. And so the FOP gene, ACVR1, also known as ALK2, acts as a kind of receptor that acts. So it has one half of it on the outside of the cell and the other half on the inside of the cell. And it acts to capture all of the growth factor signaling for one particular flavor of growth factors on the outside of the cell. And these are the BMP family, or in FOP, activin, shown by this yellow circle here. They will bind to the outside of the receptor And then it's got this, the ACVR1 gene function is to pass on that message through the cell such that it can act on our DNA and turn genes on and off. And it does this by acting as a stamp machine, if you like. So if you recognize this kind of stamp that you might have putting a stamp on an envelope on a letter, then ACVR1 is a machine in its inside here that acts to stamp like a postage stamp that sends a message onto your DNA. And this message is mediated by these SMAD proteins. So if you read some of the literature, you might read the terms BMP or activin. These are the growth factors, ACVR1 or ALK2 as the FOP gene itself, the SMAD proteins as this intermediate messenger, and then the DNA and the genes being turned on as the response. And if I simplify this into a cartoon, here we have our activin growth factor. This will be the receptor, the gene, ACVR1. It has an outside part of the cell, the inside part, and that tends to stimulate the cell to make a message to make bone. And the problem with FOP is that where we have variants of this receptor, where one of the letters in the DNA is wrong, then that has a altered message such that it's now making too much bone. And we need a way to stop that. And so some of the trials that are running, including the STOPFOP trial, they're designed such that we have a drug molecule, shown here by the triangle, which can then bind to this stamping part of the receptor to stop these messages being formed. And in the scientific world, those are known here as kinase inhibitors, because the name of this stamp machine here, technically in the scientific language, is a kinase domain. And so I've just got a little video that some of you will have seen a few times, which just shows you what this looks like in the real world. Maybe I have to take that off. Here we go. So these are all the atoms that make up the FOP protein. And then I'm just showing you how does this drug bind to the mouth of the protein. So here in the middle of the protein, we have this central pocket, or the mouth of the stamp machine. And we're going to show you how that drugs could bind to that pocket. So here's an early stage drug that we worked on with our colleagues in Harvard about 10 years ago, shown in white, and the outside of the receptor shown in this kind of mesh. And so we have different molecules that we were trying as we researched FOP that would bind to the FOP protein shown in orange here and block this site that's sending out these messages. Okay. And so an element of drug discovery is similar to Cinderella. We're trying to find what is the best shoe for the FOP protein or Cinderella. Now, there's a lot more to it than that, because we want the shoe to fit elegantly to the FOP protein, but we don't want to interfere with other biology. We don't want to, cause side effects of your heart or your other tissues. And so there's a lot of effort that has to go into making a drug both safe and highly effective. So there's some things such as making a drug that you can keep it in the cabinet and keep it store it. It obviously has to be strong medicine, like potent to stop the FOP. It has to be safe, effective. It has to be able to withstand, for example, metabolism as it runs through the body. It obviously has to stay in the body for a number of hours to have activity. You don't want a medicine that tastes absolutely awful, or else you might be persuaded not to take it. And the medicine has to be soluble, so ideally it could go through the blood. You may have a medicine that's stable to stomach acid, for example. So there's lots of difficulties in making a good medicine. And so part of our work, and this is work done by Ellie in Oxford, was to then screen what drug molecules would have all of those properties and be potentially a good medicine for FOP. And so what we did was cut off this lower portion of the ACVR1 gene product in blue here, and then screen, for example, different, as the cartoon shows, this kind of triangular drug, and see which ones of these might bind the best to the FOP protein. And that's shown in the chart in the right. And this is effectively the equivalent of a logarithmic scale. So actually these look much closer than they are, but actually this bar in purple here is binding the best with the rest red asterisks. And all of these are actually significantly worse. So we're really pleased to see a drug bind extremely well. And this drug was a drug called Saracatinib, also known as AZD0530. And it was developed in Manchester by the drug company AstraZeneca, as I think many of you know by now. And it has quite a history of working in clinical trials. So with that, we eventually went to a mouse model with our colleagues, Paul Yu and Dong Dong in Boston. And they had several mouse models, but one including the R206H physiologically accurate FOP mouse model. And in this model, you can have an injection into the hind leg of the mouse that then induces trauma such that they develop FOP-like lesions on the hind limb, giving bone formation. And if you don't have any treatment for the mouse, then you will have this FOP-like effect. But if the similar batch of littermates of the mice are also treated with Saracatinib at the same time that we give the trauma, and then after the trauma, then we can stop these FOP lesions forming on the hind limb. And importantly, we saw that the normal bone growth was unaffected. So the growth, the length of the bones was unaffected, and also the density of the bone in the legs was unaffected. And so that was highly encouraging that we could take that through from the effectiveness we've seen in mouse to see how that would work in humans. Now, we were very fortunate that Saracatinib had been used in over 700 patients in clinical trials before, including a phase 1 trial, which is there to judge the maximum tolerated dose, both as a single dose and then later as chronic treatment over time, so taking daily doses, and then also looking at the safety of the drug. Is it safe to give to humans? And that was well established. So phase 1 and early phase 2 studies located a very good dosing regimen that was safe to use. And that's from using 1 gram if you were just having a one-off dose, to lower doses of 0.175 grams a day that was used mainly in cancer as a chemotherapy for 4 to 6 weeks, particularly for ovarian cancer, and subsequently in other cancers. It wasn't effective as a sort of monotherapy for cancer. So this medicine on its own was not strong enough to treat cancer. But it's been subsequently looked at for other indications, including over 100 patients that have taken the medication either for Alzheimer's disease or for a lung disease called LAM, a fibrotic lung disease. And there, instead of just having the drug for 4 to 6 weeks, they were taking the drug for up to a year. And for this period, they were taking a lower dose of the drug. It was always felt that the higher doses were giving more drug than was necessary, and the lower the drug, though we can keep it still being effective, but at a lower dose, then you can mitigate and remove some of the risks for safety. So at the higher doses, it was having an effect on lowering blood counts, but at this lower dose, that effect is much, much minimalized. So it's less of a worry. And then at these lower doses, the main risk factor has been just starting the medication and having upset stomachs and potentially diarrhea. And so when we were designing the FOP trial, we also selected to have this low dose drug that was used for Alzheimer's and LAM, that would be the safer dose, but maintaining hopefully the efficacy. And Marelise will tell you a little bit more about the trial. But one of the things we needed to do for the trial was, although AstraZeneca will provide us with the medicine, this is an academic trial. So it's run by Richard, Marelise, and Clements, who's shown here in Germany. And so we had to write a 200-page application to the funders to try and get money to run the trial. And that was successful, funded by the EU and the Innovative Medicines Initiative. And we had to go to Brussels for an interview to justify why we wanted to start this trial. And just recapping where we were in terms of what the FOP gene does and the impact of the trials. So here we have our picture of the scheme of growth factors, the gene itself in the receptor state, stamping messages going to DNA to turn genes on for bone formation. So for the STOPFOP trial with Saracatinib, we're targeting the stamp machine itself. So this inner part of the protein, where it's sending too many signals, we just want to block those signals. Other trials, for example, from Ipsen and Insight, were designed by the same principle to target this stamp machine. Regeneron have a slightly different approach where they're taking antibody that will now act to capture and sequester these growth factors, particularly activin shown here, such that the activin can't bind the receptor and stimulate these messages. And then the earlier trials by Clementia and now Ipsen using the drug palovarotene were acting, if you like, downstream in this response area. So acting on DNA activity. So I'll hand over there to Marelise and I'll be happy to take questions at the end.
Thank you very much, Alex, for your great talk. I am Marelise Eekhoff. I'm internist endocrinologist at the Amsterdam University Medical Center in Amsterdam, the Netherlands. I am coordinator of the STOPFOP trial. It is an investigator-initiated trial, and it has been investigated already so often that it is now tested as a reuse. As you can see here, clinical trials normally follow the phases 1, phase 2, and phase 3. Phase 1 is to look for the dose ranging for safety in healthy people. Phase 2 and phase 3 is in patients. And we are now in phase 2 for the STOPFOP trial. Next slide, please. Can you hear me? Oh, yes. And therefore, the aim of this trial is to investigate the effectiveness of Saracatinib in FOP. And we are doing that by the use of the 18 fluoride sodium fluoride PET-CT questionnaires and diaries. And we are looking for safety of Saracatinib in people with FOP. And that means that we are looking for side effects and blood results. And this whole trial is aiming to discover an effective medicine to stop FOP. Next slide, please. As already said by Alex, 3 clinical study sites are participating. United Kingdom, London, Netherlands, Amsterdam, and Germany, Garmisch-Partenkirchen. But it doesn't mean that patients from other countries can't participate. Actually, different countries from the European Union patients are flying in. And we look at it case by case, whether a patient can be enrolled. Next slide, please. The study is designed as a 6-month randomized controlled trial, followed by a 12-month open label extension phase. The reason that we designed it this way is that we can compare the 12 months open label extension phase with a historical cohort, but also a group with patients who had a placebo followed by the drug phase in the extension phase can be compared by itself. What does it mean, randomized controlled trial? It means that the doctor and the patient don't know which what they will get. They can get a placebo, 50% chance, or a treatment with Saracatinib, but both are blinded, so we don't know. And then The trial will be followed by a 12-month open label extension phase, and then every patient will get Saracatinib. You are sure you will have the medication. So that is a 100% chance. And then after the study has stopped, you are allowed to go on with the medication. There's Helsinki regulations, and at the moment there are even patients already going on with the Saracatinib. But of course, we will still monitor the safety. Next slide, please. Here you can see the visits. There are 10 visits in 18 months. It does mean that there is a screening visit followed by a visit after 3 weeks when you start with medication or placebo. And then only every 3 months you come for a visit to the center. This is a bit less intense than other studies. But it is well designed, and we could design it with all the boards. And I want to emphasize that this investigator-initiated study is collaborating with different boards, and in one of the boards there are all European patients organization. They are joining, and also FOP, so we can discuss everything. That is very important. So only 10 visits in 18 months. The medication is daily oral administration, and the medication can be crushed. That's very easy to use. We don't ask that much. We ask you to fill in a diary, which is very easy, and you will get it every day. A blood urine collection only during the visits, pulmonary cardiac test on several points, questionnaires during the visits, and of course the PET CT scan 3 times and a CT scan at the end. Next slide, please. What are we looking for? Well, we are aiming to enroll 20 FOP patients between the age 18 and 65 years. Because the study is very small, we can only include patients with a classic R206H mutation. People, of course, need to be willing and able to participate. Of course, you need to fit in the CT scanner, but we can discuss. And there are only a few other health issues that are important, but most of the patients can participate. Next slide, please. Well, how far we are. The London site, but of course, Richard will tell more, is hoping to open in the next month or 2. And we are aiming to finish enrollment rapidly by the end of April. And currently, patients are in all stages of the trial. Of course, we had to deal with COVID, so we had a delay, but there are already 14 patients enrolled in December, last December, and even 4 patients are ready with the whole trial, and they want to proceed with the medication. So they are now monitored by us. Okay, what can we say about the side effects? We haven't seen serious side effects. Of course, the normal ones like stomach and diarrhea, but not important ones. And we are very happy and lucky about that. Next slide, please. Well, here you can see that we have a wide support team of people from Amsterdam who are helping us, statistician, ethicist, a monitor that goes to all the sites. And of course, we are really, really lucky and happy that we can collaborate with the governance boards, the funders, the data safety and monitoring board, and the stakeholders boards, including all the FOP patients organizations from Europe and FOP. Next slide, please. Okay, I hand over to Richard.
Thanks ever so much, Marelise, and say thank you to everyone for giving up their Sunday evening. what I'm just going to do is very quickly just follow on what Marelise and Alex have talked about, just to let you know what's happening here in the UK. So can I have the next slide, please, Alex? Thanks. So I thought it just interesting just to highlight for people, I think we're very fortunate, that we've got, a number of trials going on internationally, in FOP. Obviously, some have been sponsored and supported by pharmaceutical companies, but obviously this study that we're involved in is sort of funded by by the EU, which we're very for the sort of an academic initiative. So I think we're we're really sort of moving forward, as you've heard from Marelise, regarding the how the study is going. The UK sites, just to sort of highlight, obviously we were hit with the COVID sort of problem causing delays in sort of ethical applications. One of the challenges we've had here is just sort of getting the PET-CT, which obviously Marelise talked about that being sort of one of the outcome measure that's being looked at. So we've just had some challenges finding a site that can do that for us, but we've identified a site that can do that. So as Marelise said, we're really hoping to be ready to open for recruitment very, very shortly. If we can just build the next slide, I think it's just I'm going to just highlight things. So just let people know, obviously, our site, as we'll sort of illustrate, is we've had a lot of experience in running trials with patients with FOP. We were the main UK site for the palovarotene program, which is obviously now sort of finished. It's sort of there is no longer recruiting. We were a site and are a site for the Regeneron garetosmab study. And we will be opening later in the year for the Ipsen and the Insight study. And obviously we are an active site, for the Saracatinib study. So again, I think, and again, other sites in Europe also will have a number of trials that they are able to recruit their patients to. So if I can have the next slide. So I think, the interesting thing is, we are in a very rapidly changing sort of area at the moment. And I think, again, I mean, for you as people listening, I mean, I think, you are doing your homework, you are, starting to look and see what the studies are out there. I think the important thing is when you've got a number of studies to choose from, obviously, make sure that you have a good detailed read of the patient information leaflets that are available, and in a way, what Alex has presented regarding the background to Saracatinib will be sort of included in that sort of information, because you're going to want to sort of understand the mechanism of the drugs, how many people it's been tested in up till now. I think also, again, talk to your FOP doctor, or again, check out what IFOPA or the ICC websites have information about sort of clinical trials and studies. And these, again, are really useful resources, because I know not everyone online might have, a very dedicated FOP doctor, or the FOP doctor may not be participating in the trial, and, you're going to another distant site to get some information. So I think, again, using whatever resources you can to get this extra information is really, really important. So the next slide. So, and then just do the build, please. So I think what, again, for us as both clinicians, or as majorly for you, I mean, in a way, although these trials and studies we're hoping to find a cure or a treatment for FOP, I think the most important thing for all of us is to make sure that the medicines that we're researching are safe. And again, that is really important. And some of the governance sort of processes in place that Marelise has in Amsterdam are very good for checking the safety of the study. And obviously, with regards to efficacy, again, the study that in the STOPFOP is looking at the PET-CT, and Marelise and her team in Amsterdam, are leaders basically in understanding the PET-CT and its role in looking at people with FOP. So again, we've got mechanisms in place in this study to really make sure that your safety is paramount. And then obviously, if the drugs are going to work, we're really hoping that we'll be able to demonstrate that in this study, in the study that's happening. So next slide. So again, I think this is just a very sort of small sort of list of, what I think you need to be thinking about. And again, one of the first things, obviously, and I'm very well aware in the STOP-FOP trial, is it's a study in adults with FOP. And, we know that the majority of the trials to start with have all started off in the adult population. but we know that to really be effective in FOP, we need to move into children. And so again, that's just something to look out for with the various studies. But of course, as we start demonstrating that drugs are safe and perhaps show evidence of efficacy, there may be possibilities to move into the younger pediatric groups. I think, again, look at the various studies that are out there with regards to how long they are and how long you're on placebo. Obviously, again, as Marelise pointed out in the study protocol, it's a 6-month placebo component where you could be on the real drug or you could be on the placebo, and then you swap over, and it's an 18-month study in duration. Obviously, again, a lot of questions, a lot of patients will say to us, well, great, I'm in the study for 12 months or 18 months. What happens at the end? And again, whether the drug's available at the end of the study period is really important for you, I think, as individuals. Thinking about the formulation, whether it's a tablet, whether it's given intravenously, whether it's a liquid formulation, or as Marelise said, with the Saracatinib, it can be crushed. And again, that can be something that's important for people that have swallowing problems. I think, again, another important thing for patients and their carers is the frequency of the visits to the site. Often with these protocols, there's quite busy at the start of the study, and then as you get established on the study, the number of visits become less. But you just need to look at that. How much time commitment is it going to be for you or for your carers? Can some of the visits be performed closer to home? That might be important for people that are traveling from, other longer distances or from other countries. Are there ways? And again, I know some of the drug companies allow sort of home care, sort of nursing systems. But again, these are things that perhaps you just need to be thinking about. What sort of safety assessments are people doing? And how often are those assessments being done? Are you having certain tests done every month, every 3 months? Because again, that's going to sort of be something that again will involve time and commitment. I say most of the studies are very similar with regards to they're involving some form of CT. Obviously, the STOPFOP studies using the PET CT, but the other studies are using mainly whole body CT as well. And then the travel support, because again, we do know that coming to site in the hospitals for assessments and things, you might need to stay overnight. So again, you just need to look at what travel support is available, either through the study itself or maybe again through the local, your sort of national sort of FOP sort of charity and support groups. So again, these are just things I think you need to consider. So just very, so that's fine. So next slide, Alex, that's great. So just very quickly, just wanted to introduce our sort of team, as you probably, some of you all already know. Obviously on the right, we've got basically myself and Jude Bubbier, who's my consultant colleague, and Martin, who's our clinical nurse specialist. He's mainly involved with the our sort of health service care of patients, but is often a really good point of contact. On the far left, we've got our research fellow, Dr. Gak, who's basically now sort of working with us on sort of looking after our FOP patients, and she's actively involved in sort of recruitment and assessing your patients. And then, like every research team, there's a lot of people sort of both in the background and helping, and basically we've got a really good sort of team. They've got a lot of experience in sort of looking after FOP patients, and again are well used to sort of the dialogue that's needed. So we're very fortunate to have this sort of team available, and again, I'm sure some of you may already know some of them in your experiences with some of the other trials that you might have participated in. Next slide, please. So again, just be aware there are some contacts again that you can see with regards to sort of how you can get in contact with people, either to find out about the study itself, obviously with us in the UK, but again also again looking at the other sort of groups like FOP Friends and IFOPA will also have information about the various studies that are out there, but particularly in this context for the STOPFOP study itself. So that's basically where we are with things in the UK. So I think we're now sort of got sort of about sort of 20 minutes or so time for questions. And I think I've spotted that I don't think the Q&A function has been working as well. Is that right, Helen? But I'm not sure whether we've had any coming through, but or are we going to open it up?
The Q&A question has been working. The chat function wasn't set, but I think the questions that we were set beforehand have largely been answered by your presentations. So I don't know if you want me to go through them again, just for clarification. One was, are there any results from the phase 1 of this trial? So you explained that phase 1 was the safety element, didn't you, of a trial?
Yeah, phase 1 is only in healthy people. And in Saracatinib, it has been already investigated in many healthy, but also in other patients with other diseases. So we didn't have to repeat that anymore. We have enough safety data already. Yeah.
Then the next question was, if the medication seems to be suiting the person with FOP, are they able to stay on it after the trial has finished?
Yes. Yes,
So
they are allowed. Oh, sorry, Richard. Yeah.
Yeah, I suppose yes. I think as Marelise said, because obviously trials often have there's a staggered start. So some people will get to the end before the study has completed. So I think in that context, this study does allow people to carry on. Obviously, once the study results get analysed, I mean, if the drug works, obviously then it moves on to a next phase in the development process, like Alex and Marelise had said. Obviously, if the study hasn't worked, then obviously presumably then at that point we would the drug that component of the study would stop. So it depends. It's until the study results have been analysed, and then we know whether we're moving on to the next phase.
Okay, thank you. Another question that's just come in. If the results are favourable, because you said this was only available for people who had the classic FOP, are you going to extend this to patients with non-standard variant FOP?
Shall I answer, Richard? Or?
Yeah,
Go ahead.
you answered. Yeah.
Yes. If it is working well, what we hope, then we will go to phase 3. Then it will be a larger study, and then we also can include patients with non-classical mutation. But now, for the beginning, in such a small study, unfortunately we can only include patients with the classical. But the next step, certainly, yeah.
Thank you. Next question is, obviously we've still got to fulfill the STOPFOP trial for adults, but if it's successful and the results are pleasing for STOPFOP with adults, will you be extending it to children, and will there be the expectation of the same number of trial patients, so between 30 and 100 children to participate? How does that work if it does move to pediatrics, children?
Well, I can answer, but I think it's good that we both answer. So Richard, maybe you can answer now, and then I take the next one.
Again, I think with regards to moving into children, I suppose the first thing would be we would probably need to just go back to some of the more basic science, because I think from what Alex presented with regards to some of the preliminary stuff and also where the drug had been used, I'm not aware yet that we've got a lot of good and even that phase 1 data in children who haven't got FOP. So we would need to get some data from either some academic research or even going back to AstraZeneca to find out if they've got any data in sort of children or in pediatric, in sort of young sort of animal models, and then extrapolate and then start moving forward from there. Because I think it's not just a matter of finding that the drug is effective in adult population and then moving straight into children. I think we would at least need to do some more basic science to at least start looking at some of the pharmacokinetics as in how the drug is handled.
Maybe expanding on what Richard said, the key issue is safety. And so we can say a drug is safe for adults, but of course there are different conditions for children in terms of growing bones, development, etc. So there's a higher level of safety stringency that regulators look for before starting trials in children. And so we have to show that the drug in all the preclinical models has that safety required.
Okay, thank you. Then it says, someone's here asked, since we've enrolled you've enrolled 14 people on the trial so far and your trial size is 20, are you looking to enroll 6 patients at this time? Are you looking for 6 more patients?
Yes.
Yeah. And so anyone in the UK or anywhere, so it's the UK would go to London probably, yeah? And they'd contact Richard and the team, and anyone who's listening in from Europe, they would also be able to get in touch.
Yeah,
Absolutely.
I mean I mean they could go to they could go to Marelise's site, they could go to Clement's site in Germany, they come to England once, you know, so anyone can do that. Yeah.
Okay, and also, will there be any help? I think you did touch on this. Will there be any help with the cost of traveling and staying overnight? So towards the cost of the trial, so participating in the trial, I should say, for patients.
There are some costs included again, but again, Marelise knows more how that's worked with the other people visiting the other sites.
Well, we have costs. We can reimburse costs, travel costs, but when patients are coming from far, we also try to collaborate with the patients' organizations and to find other options to reimburse, because the study has a fixed budget and we are already going on two and a half years longer due to the COVID, but we can discuss with the patients and we look for options together. Yeah.
Okay, thank you. And when a person with FOP appears to be in a flare cycle, so having one flare up after another, do you think or have you got any, ideas that the Saracatinib is going to be able to help stop the flare cycle?
Well, yes, but yes, that's what we are aiming for, and that we hope to get this result. Yeah.
And I think someone's asked, this isn't relevant specifically to the STOPFOP, but they've been contacted last year. A patient was contacted last year regarding the Falcon trial, but they've not heard anything further. Is this because they have the non-standard variation of FOP?
No, if this was a UK patient, and I think I know who it might be, but from the question, but I think it was mainly because, again, one of the safety measures that was needed for this study was a heart sort of echocardiogram, so like a heart scan, and again, there was just some issues about finding a site that could do it, because sometimes it can be more challenging. So there's been a bit of a delay, but the Falcon study will be and again, there've been some things going on, I think, with regards to just data analysis that they were doing in the first number of patients worldwide. So there's the pause on recruitment. So that won't now probably start recruiting until the end of February. So anyone that's been contacted about Falcon will be recontacted once the study basically is up and running again. But there's been a pause now internationally whilst they just look at some of the preliminary data to check that it's safe. And again, as we know, safety is really important. And then if they're happy that it's safe, then they're going to go into the next phase of recruitment. So yeah, so it wasn't, I don't think it was stopped. People weren't contacted because of not being a variant for that, because of not having the classic mutation for that study.
Okay, thank you. What do you anticipate to be the size of the phase 3 trial once you get to that point? They say 20 people enrolled in a study is a very small group. Can you share why you felt you would get indications of efficacy with such a small number?
Well, we have strong data, preclinical data, of course. We have a design where we can have 2 ways of comparing the results. We hope that this trial with 20 patients will give us an answer for this medication. And maybe Alex or Richard, you might add your ideas.
Yeah. I guess we don't want a drug that will just make a 5% difference. We want a drug that will make 90% difference, that would really, or 100% difference to be ideal. So they would really impact and prevent FOP. So when we want a really effective drug, then if it's truly effective, we should need fewer people in the trial to show that. And that's mirroring what we saw in the mouse models.
Okay, thank you. Not quite sure. And this one says, how can one continue to get the drug after the end of the trial? Would would then you have to take part in a follow-on trial, or can you just be allowed to continue just to take the medication?
No, we have to take care of the safety. So we have a follow-up study, and we can only give the medication till we really know the efficacy, and when all results are known, and then we can make the next step. But till then, you can use the medication, and AstraZeneca is supporting us in that. Yes. But we are still monitoring, but because safety is the most important thing there is. Yeah.
Sorry, did you want to add something, Alex?
No.
No. Okay. Are you able to start the trial whilst having a flare-up? If you're currently in a flare-up, are you able to enroll on the trial?
Yes, but you have to understand that you don't know in the first half year of the trial, but all trials have the same start, that you don't know whether you will have the medication or the placebo. So you still need medication, but you can start definitely when you have a flare-up.
Of course, there's a practical issue of whether you want to travel to a site when you're not feeling so great. But apart from that, yeah.
Yeah, that's true. That's a good
yeah, very
answer.
true. There's two questions come in that are very similar now, which is if you are already on another trial, so if you want to go join the STOPFOP trial and you've either previously been on another trial or you are currently on another trial, can that be organised? Can you switch over? And what's the typical time frame for this?
So yes, you can swap. Normally you have to give a certain number. You have to obviously allow the previous drug to wash out of your system, which is probably about a month or so. But we would just need to look at what trial you'd been on, and then one would be able to do that. So yes, I mean that is I think we know that obviously in the patient numbers with FOP are quite small, and people that might be able to come to site to have a try, be in a study. So therefore we know people already might have moved from one trial to another trial already, and it will be the same here. So it is possible to do that.
Okay, thank you. Sorry, can you just clarify what the youngest participant age is for the trial? 18.
18.
Yeah. That's just been asked again. Thank you.
Yeah.
I think, does anybody else have any more questions? Or I think that's all the questions that I was given prior to the webinar and are in our Q&A section at the moment. No, I've not got. Oh, hang on.
What is the follow-up duration?
Yeah, that was it. So that's the last
so
question.
Marelise, the follow-up duration. So how is it basically? So once they finish, how long is the follow-up? Is it sort of? Have you got a?
It's till the end of the study when we really know all results and how the study will move on. And after we will have 3 monthly contacts, mainly by telephone, but also visits, but less intensive, of course. Yeah. We still go for safety and we still be monitoring. Yeah.
Okay. I think that's all our questions then. I think. Thank you. I don't know if you've got any couple more slides. There's a couple more slides left. I think maybe the logistics of London, maybe. I
think I deleted some of those.
Did you leave them there? There was at the end of my chat. Right. Well, in which case then, if there's no more questions, oh, hang on. Oh, someone said, we love you all. There you go. I feel like I should just leave it at that point. I just want to thank everyone, all the people who joined in with the webinar this evening. Thank you for coming this Sunday evening. A big thank you to Marelise, Richard and Alex for giving up their time on a Sunday evening to support our FOP community, both today and all day, every day. I'm sure Richard knows what I mean. And also, I don't know if anybody who you've seen, but Michelle from the IFOPA is hiding in the background. And a big thank you to both Michelle and Hope for helping me with the logistics of this, because I certainly couldn't have managed without Michelle today. So a big thank you to Michelle as well, because it's her Sunday afternoon. And I think she's got a big game to go watch on TV with some delicious baked goods. So I think, if there's nothing else, I think it's been really appreciated, and the webinar will be available at a later date. So thank you very much.